Awesome. Thank you, Tara, for the introduction. Today we'll be talking about the response of lichen fragments of different sizes on various substrates in a greenhouse setting. It's a little bit about the background of all of these projects, so the lichen project in general. So our overall goal was to evaluate the feasibility of terrestrial lichen transplantation in forest harvest areas. So this research was funded by the Alberta Regional Caribou Knowledge Partnership, so ARCKP, which was formed by the Government of Alberta and the province's forestry sector to find on-the-ground solutions that balance both forestry activities with woodland caribou conservation. So that funding lasted from 2021 to 2023. So we were able to find some additional funds through the Natural Sciences and Engineering Research Council of Canada. So that's NSERC, which helped us continue the research from 2023 to 2025, which was really good because then we got additional data, especially it's really important because the lichen grow very slow. So we got lots of data from it. So we really would like to thank both partners for funding this research. So as I said, overall goal was to evaluate the feasibility of terrestrial lichen transplantation in forest harvest areas. So some of our objectives also included, sub-objectives included examining the effects of substrate. So how do these fragments do on mineral soil, moss, pine needles, and woody debris? And also another sub-objective is examining the effects of fragment size. So are small, medium, large better for survival and health? So we conducted a series of studies. So I've highlighted them here. So the first thing we did was conduct a literature review. So we wanted to understand what's been done on this topic on restoration using lichen previously. So we scoured the literature through different platforms, and we actually, this is published on the ARCKP website. So the link is under number 1. So we also presented a webinar on this on May 2022. So this is actually available on the FRI research channel as well. So you can take a look if you missed that webinar. Our second objective was to assess historical lichen transplantation sites. So how were historic sites doing like 20 years later? So Michael presented on that topic, July 2025. Again, it's also, the webinar is available on the FRI research channel. You can access it through the link provided. So the third study was to examine the terrestrial lichen responses to fragment size and substrate in a greenhouse setting. So that's today's topic. Another study was looking at terrestrial lichen responses to fragment size and substrate, but not in a greenhouse setting, in the field setting. So how did they do in the field? So the link is also available to Michael's webinar on this topic. I think it was also Jean-Marie and Dua who presented on this. So this was presented September 2025. So if you are interested in the previous webinars, please just request for the links and we can also provide it to you. But today's focus is looking at responses in the greenhouse, and then Michael later on will talk about how this the greenhouse results compare to the field results and what we see. So to start off, lichens play a very important role in the boreal ecosystems. They help fix carbon and nitrogen, and they help stabilize soils. They serve as food for caribou, especially during the winter time, where there's not very many food available at that time, and also other animals. They should not be neglected in reclamation efforts, especially if we're trying to achieve that equivalent land capacity. So we want to make sure that the ecosystem looks the same as what it was before the disturbance, and that includes lichen and mosses. So greenhouse studies, what research has been done on it? So greenhouse studies may help us understand the best transplantation methods. So if there's a good result in the greenhouse, maybe we'll try that in the field. So the growth of lichens in a greenhouse environment was first observed in 1963 in Science by Culbertson, but not very much greenhouse studies have been seen since. There was an informal study by Stewart in 2019 where they grew Usnea and Ramalliana species on sterilized branches in a greenhouse. So this was not a peer-reviewed paper. The lichens died within 2 weeks to 19 months. So there was quite a range. Um, so yeah, as they see in the abstract, discovery of the first species of lichen capable of tolerating greenhouse conditions. They do mention that lichen is a plant, which it's not. So it's a fungi that has association with algae, sometimes cyanobacteria, and also in some cases yeast. So we were interested looking at the effects of substrate. So what we found in the literature was that the growth of C. arbuscula, which is also mitis, fragments were greater on moss compared to mineral soil, twigs, and barks in clear-cut areas. But for some reason, there was no difference in substrates in a pine stand. So maybe that was due to the cover and the reduction in evaporation. So many reindeer lichens do not appear to grow very well when placed directly on mineral soils. Fragment size we were interested in because we found that it might have an effect in the literature. So fragment size, portrait role in transplant success, we're going to try to find out. So Citraria islandica lichens planted in mats had higher growth over time relative to those dispersed by fragments. So if you take a huge clump of it, it seems to do better than when you just throw out fragments. Sean Rapai, one of our collaborators, found no significant differences in percent cover for various lichen species when transplanted as fragments, mats, or even a hybrid. So that was very interesting in that study. That's actually a photo from one of their sites. So some common terrestrial lichen species. So these are the reindeer lichens. So they include Cladonia arbuscula, which I'll be referring to as C. mitis, C. rangiferina, C. stellaris, and C. stygia. So these are our classic reindeer lichen groupings. But terrestrial lichen species also include other lichens. So we have Uncialis, we have Centraria atlantica, Flavocentria nivalis, and Gracilis. So those are on the top. We have our dog lichens on the bottom, so Piltigera malaceae and P. aphthousa and Leucophlebia, and also our Sterocolon tomatosum. That one actually really likes mineral soils, and I think it's considered as a coral lichen because it looks like coral. So some of our methods, so we collected lichen and substrate in Peace River and Lac La Biche. So what we did was we placed 1 by 1 meter quadrats within areas rich in lichen. We identified the species within that quadrat and we collected all of the lichen within that quadrat. So we made sure to be responsible in our harvesting. So we only selected a few quadrats in a particular area and we moved to a different area just to make sure that we leave most of it behind. So the substrates, the mineral soil was collected from the top 20 centimeters of the surface. Moss was usually comprised of a mix of species, but was heavily dominated by feather mosses. Pine needles were collected from the ground, and woody debris mostly composed of bark or smaller wood chips that were degraded or decomposing. So with those lichen, we brought them into the lab, we air dried them and cleaned them. So we wanted to make sure that there was no other material in it. So we didn't want mosses as part of the lichens. They're fragmented in small, medium, large fragments. So what you see in the diagram, so you have 0 to 2 centimeter. For small ones, medium ones were 2 to 4 centimeters, and then large ones were 4 to 6 centimeters. So for the experimental plots, we had 4 substrates, 3 fragment sizes, making up a total of 12 different treatments, which were replicated 6 times. So in the greenhouse, we had 72 trays. So these trays were large black plastic trays. So they were 56 centimeters by 56 centimeters and a height of 7 centimeters. So later on, I'll show you kind of an example of one of these trays. So they were randomized across 2 benches, and 40 grams of dry lichen were applied. So we this is why it's important. We want to make sure that they were clean, was because if there was moss or twigs embedded in the lichen, then that would just add weight so that we actually have a lower amount of lichen. So we want to standardize the amount of lichen that we're adding to each one of the trays. And the photo is actually so all of these are large fragments. A is the fragments on mineral soil. B is the ones on pine needles. C is on logs and woody debris. And D is on feather moss. Greenhouse conditions. So we misted once a week from November to March. So during the winter time, this was because they were kind of in dormancy. They weren't using as much water. We increased it to 2 in spring, so March to May, they're starting to become more active. And then in the summertime, we watered a lot more from June to August because there was a lot of evaporation in the greenhouse and they were actively photosynthesizing. So we use type 2 deionized water because our water was very heavy in calcium. So that has been attributed to the decline in health of lichens in previous studies. So it's better if you're growing lichens in a greenhouse, make sure you use that deionized water. Supplemental lighting was required, but in the wintertime, we allowed for natural dormancy and we shut off the lights and the temperatures were cooled down. Measurements. Our whole experiment was started in 2021. So this was considered as time 0. We marked 20 fragments within each of the trays. And then we followed those fragments over time. There was more fragments in the trays than 20, but we just picked 20 in each one of the trays. At a later date, we assessed for baseline length and biomass. So this was March 2022. So this was after a period where they were able to acclimate to the conditions. So we didn't measure right away. We waited a little bit after we set up the experiment. Subsequent measurements were conducted approximately 16 months after time 1. In July 2023, we referred to this as T2. So we're mostly comparing time 2 to time 1, see what happens relative for growth, biomass, and chlorophyll fluorescence. So it's concluded 20 months after T1. So the experiment lasted about 2.5 years in total. So in the photo, we see, so we used a scale to measure dry lichen biomass. And on the right, what we see is measuring the longest length of the fragment using a digital caliper. So these lichens were air dried for 2 weeks prior to measurement to standardize water content. We also wanted to assess visual health. So we made a scale, so 4 being the most healthy and 0 showing signs of decay, 1 not being great, showing significant signs of bleaching. In these photos, they actually look pretty good. The left one, you see some bleaching at the lower ends of the thallus. That's typical because it's usually closer to the ground and then it's quite green at the top. So we would have scored this relatively high. So it would have been 3, 4. And with the pilchard on the right, some signs of decay, but there is nice green pigmentation. So it'd probably be a 3 for us. Same person scored from the beginning to the end to make it more consistent. So to measure survivalship and health, we used chlorophyll fluorescence. So we measured something called FV over FM. So if it's above 0.6, we considered it healthy. Below 0.6, it's stressed. These are based on plants, so it may differ slightly between species, but it's not really documented what the levels, the set points for the lichens. So 0, we classified as dead. So that was very useful. So we were able to determine survival based on that. So 0, dead. Anything above 0, still alive. For those lichen that were still alive, we were able to determine the mean FV over FM of those lichens. So for the lichens that survived, what was the health of those lichens. So results and discussion. C. Mitis was the most abundant. So it made up about 40% of all the fragments that we put out. The proportions did not significantly differ across the 12 treatments, which was great. The only one was C. uncealis, only differed in 2 treatments. But overall, the distribution of species in the different treatments was good. Visual health scores. So at time 1, so at the very beginning, our first measurement, we see that a lot of them scored a 4. So they're very healthy. But at time 3, so a later date, 16 months afterwards, they shifted towards a 2, which was a little bit concerning. So something in the greenhouse did not appear to be helping them grow and survive. So there was something about the conditions that were off for them, that their visual health was being reduced. So we pulled the data together. So we disregarded species as a factor. So we combined all of our data together. And what we found was that survival was significantly lower on wood when compared to the other 3 substrates. There were no fragment size effects. So what we think is that the lichens on the logs dried out very quickly in the greenhouse. And these responses, so when we did look at the species breakdown, so we analyzed it survival by species, what we found that the overall response of the pooled data was driven by C. mitis and stellaria. So in the graph, what we have on the left is our substrates. So what I've circled is woody debris. What we see is that it is much lower than the other groups. So survival in that woody category was not great, and it was significantly different than the other substrates. Whereas for chlorophyll fluorescence, so once again, pooled the data all together. We didn't care about species. We excluded it as a factor. And what we found was that there was significantly higher FV over FM values were observed when fragments were on moss relative to wood and soils. So no fragment size effects once again. So fragments that were alive seemed to be relatively healthy. And that overall response seemed to be, so we then went and analyzed by species. And then what we concluded was the overall pooled data was likely driven by Cladonium mitis. Not surprising because it was the most abundant in our experiment. So we kind of expected to be skewed towards the response of C. mitis. In the figure, the left-hand side, what I have circled is feather moss. So what we see is it has way higher FV over FM values than some of the other groups. So relative change in length, we pulled the species data again, disregarded that species information, and all mean changes in length were negative. So everything appeared to be fragmenting. Though wood fragmented less than the other groups, the other substrates, and small fragments were less likely to break apart, which was interesting. Moss typically, so what we think is moss typically held moisture for long periods and resulted in decay and fragmentation. However, the fragments that survived in the moss treatment appeared to be relatively healthy. So that was an interesting result we did find. And fragmentation likely was due to moving the lichen around during measurements, moving the trays around, and larger fragments are also more likely to fragment because they have greater surface area, more points to break apart. And that overall response was likely driven also by C. mitis, our dominant species, and our change in biomass. So we had our pooled data once again. So as opposed to changes in length, relative changes in biomass were on average positive. So everything mostly seemed to be gaining weight, with one exception, the feather moss group. But the change in biomass on moss was significantly lower than on soil. So that's what's circled. So what we have in the circle is that mineral soil seemed to do the very best, and our feather mosses were lower. So we do attribute this to adhesion to substrates. So the mineral soil seemed to attach to the fragments in the mineral soil treatments, which added weight. So when we did weigh them, we didn't wash them through water because then everything would have to be dried again, and then that would take a lot of time. So we tried to scrape off as much dirt as possible or soil, and then we did our measurements. Alternatively, we could have soaked them and then re-dried them again, but that would have taken over 4 weeks to process a weight measurement. So this response was not driven by Midas. It was actually driven by Stellaris and Stygia, which was interesting. So overall, we found species responses were not consistent, not surprising. So we did find some very kind of variation in responses. So small fragments of C. Midas had higher Fe over Fm relative to large fragments, while small fragments of Peltigera had lower Fe over Fm on soil relative to large fragments. So that was just kind of a contrasting example of a contrasting response. Responses were mostly driven by Arbuscula, so C. Midas again. And overall, lichens didn't seem to do very well in the greenhouse. It was kind of apparent in the visual scores. And yeah, so I'm going to hand it over to Michael. So he would talk a little bit about, he'll put all the projects into context and also compare our greenhouse study to kind of our results in what we saw in the field. So take it away, Michael.
Cool. Thanks very much, Ricky. So hello, everyone. I'm hoping you have enjoyed our discussion of the greenhouse. So yeah, I'm gonna kind of try here to mesh the results from all of the study work that we did into something that can be practical and something that we can use to drive our progress forward on using these lichen transplantation techniques. Ricky, I'll need you to forward the slides for me here. All right, so the first big picture question that we wanted to answer was, of course, can these transplants, transplanting or fragmenting lichen, can it restore caribou habitat? And so when we looked at the historic reassessments that we did, which is kind of the best judge of whether or not this is going to be able to work long term, we found that transplants were growing well after 24 years in cuplocks in West Central Alberta. And we did 2 reassessments in BC, kind of 6 and 7 years old in burn sites, and both of them were doing well after that. So obviously they're doing well after a long period of time. In our field plots, we figured that the lichens were doing quite well after 3 years with about 65 to 70% survival, and those that survived were in really good health and seemed to be growing in our studies near Fox Creek. And then just looking at the literature review, we've seen similar positive results in many other studies. So yes, we can use reindeer lichen transplanting. All right, so the next thing that is this important thing to think about, as you guys that are listening are probably a lot of forestry related people, obviously, and so is this important? Do they need to get transplanted, or is this something that nature can take care of on its own? And so a lot of these studies had controls out there to see, are the lichens coming in on their own? And so that really helps us to answer this question. So in the 24-year study, there was a small amount, very small amount of one species, Cladonia mitis or arbuscula, that was coming back in a little bit in the controls, but in the burns sites, there was nothing. And in our Fox Creek trials, it was after 3 years, but still there was no natural ingress of any reindeer lichens. As far as them dispersing on their own, we know that dispersal is is one of the challenging thing for reindeer lichens. And in the 24-year cup study, we did test to see if there had been dispersal from the plots, and we found basically almost no dispersal, just a little bit of growth outside of the plots that were there that had been successful, and that was about it. And I would encourage the forestry-minded people here to think about reindeer lichens much in the way you might think about spruce trees, in that white spruce trees are, and like reindeer lichens, are both kind of slow-growing species that that are slow to disperse, take a while to get there, and yeah, grow slowly once they're present. And so if we want to draw a kind of comparison to trees and lichens, the reindeer lichens are kind of comparable to spruce. And so if we want reindeer lichens back to restore caribou habitat, just like we would plant spruce trees in our cut blocks, our harvest areas after we've harvested, to restore areas for forestry purposes, if we want to restore areas for caribou purposes, caribou habitat purposes, it seems that lichen transplanting would be the way to go. So yes, these transplants are quite important if we are to maintain caribou habitat on the landscape. So the next question is, how could we possibly do this? What's the best way to plant or disperse them? So we found that in trials that planted clumps and trials that dispersed fragments, we ended up with similar covers in both the 24-year trials in Alberta as well as the trials in one of the trials in BC near Messalinka River. So yeah, they maybe have slightly different biomass that wasn't compared, but overall can produce similar coverages. So trying to transplant chunks of reindeer lichen is challenging. It takes time, whereas dispersing fragments is quick and easy. So probably that's going to be the more cost-effective way if we are to implement this over larger areas. In Tedichuk Lake area, they did their trials dispersing fragments by hand, by leaf blower, and by airplane, and they all had similar success rates. So that was great to know that it doesn't really matter. The leaf blower apparently didn't work very well. It was a lot faster to try and just easily disperse them by hand than trying to shove them in a leaf blower where they would clump together and stuff. But we tried in one of our other trials to see if we could use hydroseeding to disperse the lichens more quickly and effectively. And it did work to disperse them, but they did not have great survival. There could be some ideas to try to refine that technique more to increase the survival, but that would need to be done before we'd want to put hydroseeding as a large-scale strategy. So over small areas, dispersal by hand is mostly going to be we're thinking is the most cost-effective way. But if we're doing large areas, we could think about some sort of aerial dispersal. Was effective in Tedichuk Lake, but they did note that it was quite expensive. But once you have that sort of scale, then doing it at larger scales, then that might drive it down to a more reasonable price. Another thought that I've been kind of hoping to be able to pursue is trying to see if we could disperse them with drones. The dried fragments are quite lightweight, and so that might be a strategy that could work, but would need to be trialed. Another interesting finding is that we shouldn't do it too densely. The 24-year trial showed that we had some competition happening, or at least some good evidence of competition, especially where we dispersed fragments or they had done that in the plots. And another study that just came out last year showed that when they dispersed 14 grams per meter squared in some plots, and then 70 grams per meter squared In other plots, after 10 years, there wasn't any significant difference in cover. So it would make way more sense to make better use of your source material and go with a lower coverage, closer to the 14. Next thing is, how do we deal with the fragments? Do we break them up into lots of tiny pieces to spread them out widely, or do we use larger pieces? So we did find that the small fragments did not perform nearly as well. They had lowest cover and survival in the Fox Creek trials and lower health, at least for some species. In the greenhouse, as Ricky has just talked about, the small had the best change in length, but other results were variable by species and what we were measuring. Overall, the medium fragments seem to have the best coverages, especially in the Fox Creek trials we had that, and the larger fragments, we had higher survival in our trials. And in the greenhouse, we found that Rangiferina, the largest the best survival was with the largest one. So overall, then we could say if we want to spread it over a larger area, the medium fragments might be the best. If we really want to make sure every fragment survives, then large fragments might be the best. All right. And the next thing is, which species should we use? Should we be very fussy about which species we select? So the great thing is that it looks like all of the common reindeer lichen species that are out there seem to work well and respond well to dispersal and fragmentation and dispersal. So the little letters that we have after here are having to do with which studies utilized and found that those different species performed well. So the Cladonia arbuscula / mitis, which is by far the most common reindeer lichen in the boreal. We have lots of good evidence that it works really well. Rangiferina, slightly not as good, but still quite good performance. Stellaris performed really well. Uncialis performed really well. And then the stereocolumns in both the BC and our trials, it performed really well and had really good survival. And that's kind of an interesting one because that one is one that also has a cyanobacterial partner, and so it's known to fix nitrogen, so it could help with nitrogen fixation and enrichment. We did find that the black-footed reindeer lichen, Cladonia stygia, did not do well in our Fox Creek trials, and in the greenhouse, the Peltidras didn't do very well. So those are probably not as important ones. Stygia is quite a rare species of bogs usually, but so it wouldn't be one that we'd have to like try to select against, but we wouldn't want to prioritize it either. So yeah, so there's arbuscula, Rangiferina, and Stellaris working well, Uncialis and stereocolon working well, but not mitis or Peltidras. Right, so the next thing is like, what kind of habitats, what kind of environments should we be putting these in? So all of the historically reassessed sites, they were in sites that would naturally have a good amount of reindeer lichen cover. So they were mostly xeric to sub-xeric nutrient poor ecosites. So we have the best support for those as far as long-term success. Our Fox Creek trials were in a little bit moister and a little bit more medium nutrient sites. They're kind of more subhydric to subhydric. And they still had decent success, but not as good support for like long-term survival. And so then I've got a few other features that were things that we had either observed or been able to statistically show had better survival. So in sites where there are some lichens, more lichens will do better. So they're not like specific that one lichen species will do well on one site and one will do better on a different site. It seems like a good site for one lichen is a good site for most lichens. We had better success on lower slopes. That might have just been a fact that they didn't fall out of plots as easily, but overall lower slopes were better. feather moss. A lot of feather moss didn't seem to correlate in our Fox Creek trials as well. Interestingly, forb cover was something that we had. Different studies had opposite results. So in our Fox Creek trials and with some lichens in the West Central Alberta's trials, we had lower forb cover correlated with better results. But then in other species in the West Central trials, higher forb cover was associated with better results. That's probably having to do with in some ecosystems, then we have uh, drier situations that support some growth. Maybe that's where the lower forb cover corresponds with better growth, and higher forb cover in more moisture, nutrient rich. This is kind of my thoughts based on observations. Then perhaps it's too much shade for them. In the West Central trials, we also found that having too much woody plant cover was bad for the lichens. So we would want to have trees more spaced. We would not want high density plantings going on with these lichens. Not an overly dry microsites. The greenhouse wood showed that, and some Fox Creek ones showed that the very super dry microsites were not great for them. And then just observationally, we saw lichens that were directly under younger trees tended to get smothered by the needle fall, and probably a lack of light as well. And in our Fox Creek trials, we found very poor success in our lower damper areas, but again, that's kind of harder to quantify, so we didn't have statistical results to support that. So then what's the next steps if we want to start implementing this on larger scales? Um, so obviously we know that this can work now. We know that it's a viable strategy and it's an important thing to do. So larger scale trials are what we're recommending as the next step in this research to demonstrate the cost at scale, and then we can also have larger areas where we can throw more plots and better understand what site variables correlate to better or worse outcomes. The other thing that's really important is just how do we harvest the lichens? You don't want to kind of rob Peter to pay Paul. You don't want to muck up one ecosystem to try to restore another. So that would be a great thing to do next, is just to see how best to harvest your lichen source material and monitor the recovery of their source sites. And then another interesting thing that is not shown well by the literature yet is just looking at that relationship better between woodland caribou and reindeer lichens. There have been some studies that have kind of supported the idea that woodland caribou are major dispersers of reindeer lichens, but there's not a lot of great support for that, not a lot of study that when I've dug deeper, not great support for that. And if reindeer like woodland caribou are major and important, especially long distance dispersers of reindeer lichen, then it would be incumbent on us as people working on the landscape, that if we are contributing to and trying to help with the issue of the drop in population of woodland caribou, then they aren't doing their job dispersing the lichen because their populations are so low, and this may be contributing to a loss of habitat for the caribou because the lichens are not getting to the sites that they would normally get to. But again, that's another really interesting area of study that would be really cool to research. So again, Ricky acknowledged them at the beginning, but I'll just mention again, thank you to the ARCKP for their funding and FRIA, and of course to NSERC who supported our work.